So the previous video reviewed the anatomy of the heart, and now just a few thoughts on its physiology, the cardiac cycle, and then a few remaining topics. So first off, muscle, under our normal experience, doesn't do anything unless it's told. So my arms don't have to I don't have to worry about them flailing around unless the brain tells them to. So my biceps will not have electrical activity unless my brain and spinal cord send it the signal. But cardiac muscle has about 1% of its fibers which are different. They are what are called autorhythmic. I don't know why you're being difficult. In that they go to action potential on their own. So without the brain and spinal cord, I'm sorry. I don't know why I was freezing. So without the brain and spinal cord sending it a command, these fibers go to action potential on their own. So that means that the heart establishes its own rhythm. So out of nowhere, the SA node will fire. This is what we call the primary pacemaker of the heart. So out of these areas which are about to appear in yellow, one of them fires first. It then sets the pace for the others and is known as the primary pacemaker of the heart. It is known as the Sinoatrial Node because it occurs on the wall of the right atrium. But as I will explain in just a few minutes, there's a chamber in fish hearts and in the embryos of human hearts called the sinus venosus. So this area starts off in the sinus venosus. That's where the S comes from. But then as that fuses with the right atrium, it becomes part of the atrium. So this area of the heart says a new heartbeat will begin now, not because the brain or spinal cord said so, but because it says so. The message gets to the atrium, and then we get to this area known as the AV node. The AV node delays the message. So by this point, the atria have received the message, but the ventricles have not yet received the message. And for that reason, when the heart contracts, it contracts as two units, the atria, then the ventricles, because the AV node delays this message. From there, the message will spread through the AV bundle to the right and left bundle branches to these Purkinje fibers. And together, these areas make up what's known as the intrinsic conduction system of the heart. These areas are autorhythmic. They will go to action potential on their own without the brain or spinal cord having told them so. Now, they are so very important in the physiology of the heart, so that if you were to ask what is the effect of a heart attack, it varies. If a heart attack interferes with this intrinsic conduction system, it is far more significant than if it didn't. And so then we see electrical activity in what we refer to as the cardiac cycle. And so in this electrical activity, there's nothing happening, and then all of a sudden there's a blip of electrical activity, and then we go back to the baseline, and then another blip of electrical activity, baseline, and then another. We can name these areas as the P wave, the QRS, the T wave, and they signify different aspects of cardiac physiology. So the atria, they're involved in the first half of the cardiac cycle. The P wave marks where the atria depolarize. They start their electrical activity. And in the QRS complex, they repolarize. They end their electrical activity. And in this time period here, the atria then are in systole. They're contracting in this area depicted by the green box. The ventricles, they are then active in the second half of the cardiac cycle. They depolarize in this region here. They repolarize in this region here, the T wave. They depolarize in QRS. They repolarize in the T wave. And this area in green here, that signifies their systole. So in a heartbeat, the atria, they are in systole for much of the first half, from partway through the P wave to partway through the QRS complex, and the ventricles in the second half from partway through the QRS complex to partway through the T wave. And so in a cardiac cycle, once again, it begins when this primary pacemaker in the SA node fires. From here, messages get throughout the atria and are delayed at the AV node. That's why the atria are active in the first part of the cycle. And it's their depolarization as they begin their electrical activity. That's what we depict as a P wave. And then as they contract, this is what is known as systole. This occurs from partway through the P wave to partway through the QRS complex. The QRS complex is when the atria end their involvement, so they repolarize. And that then is when the, I'm sorry, let me go back. The AV node delays the message, so the atria get it first, but the message will then go from the AV bundle to the bundle branches to the Purkinje fibers. Once again, these yellow areas are the intrinsic conduction system of the heart. They are autorhythmic. Then starting in the QRS complex here, this is when the ventricles are now aware that there is a new cardiac cycle. So their muscles can depolarize, and a split second later, this ventricular muscle then begins its contraction. And so this cycle repeats itself every heartbeat, and we could get more involved, and in future coursework perhaps you will, because you can tie in what the valves are doing. You know, they are open when blood attempts to go in the right direction. They close when blood attempts to go in the wrong direction. And so, for example, when the pressure of blood in the atria is greater than the low pressure in the empty ventricles, this pressure will push the AV valves open, causing the ventricles to fill. So valves open and closed because of pressure. When a little later in the cardiac cycle, the ventricles are squeezing, now blood will try to go the wrong way from the high pressure ventricles to the atria. And that's what causes the AV valves to shut. When valves shut, there's turbulence in the blood, a swirling, and this then results in a heart sound. And so during a cardiac cycle, we have a lubbed up. The first heart sound, the lub, that is the turbulence in the blood when the AV valves shut in the QRS complex. In the same way, the semilunar valves open when the pressure in the ventricles is greater than the pressure in the vessels. And this happens in the second half of the cardiac cycle. And thus, as the ventricles squeeze and squeeze and squeeze, we can have during the ST segment the ejection of blood. When the ventricles are then done their systole, we have a higher pressure in the vessels than in the ventricles. And so then blood would then try to go back into the empty ventricles. This is then when the semilunar valves would shut, and this is when we get the second heart sound, the dub. And so there's a number of events in the cardiac cycle, because one can worry about electrical events, whether the muscles are in systole or diastole, whether the valves are open or closed, where the blood is, etc. So it's complex. There's a lot that goes on at one time. As far as other topics, just the idea of, while the heart is a pump, it is a smart pump which can vary its cardiac output. So the amount of blood that is being pumped in a minute, which one could get by multiplying your heart rate, the number of beats per minute, by what's called the stroke volume, the amount of blood coming out of a chamber in any one heartbeat. When one multiplies those two numbers together, you get how much blood the heart is pumping in a minute. But the heart is a smart pump. This can vary. So during exercise, for example, we are pumping more blood per minute than we do at rest. And so therefore, one of the aspects you would consider in the heart is what changes the cardiac output. So we can change how much blood comes out in one heartbeat. We can change the stroke volume, but then we can also change the heart rate. So for example, the nervous system sends nerves to touch the SA node and the AV node, and this encourages them to speed up. The hormone epinephrine, which is released by the adrenal glands over the kidneys during an emergency, that encourages the SA node to fire more quickly. And other things like warmer temperatures, higher calcium, all of these can then increase heart rate. And so while the heart is a pump, it is certainly a smart pump which can vary its cardiac output. The blood is the fluid which is pumped. The heart is what pumps this fluid, and then this fluid is then pumped through blood vessels. We have two big types of blood vessels, the arteries and veins. Now they have the same three layers. They have the outer tunica externa, whose collagen fibers help to anchor it, the smooth one cell thick lining of the tunica interna, or endothelium, which lines the lumen where the blood is. But then there's also a middle layer which can have muscle. That's the tunica media. And this smooth muscle can undergo vasoconstriction, which would make this blood vessel narrower so that less blood would pass, or undergo vasodilation, so it would become wider and more blood would pass. Arteries are carrying blood away from the heart. Veins are carrying blood to the heart. And because arteries have thicker walls than the veins, one can identify them under the microscope. So notice here, the artery has a much thicker wall than the vein, and it's easy to identify. Once, say, your left ventricle pumps blood, this blood then travels through what are called elastic arteries, which have elastic tissue in their wall, to the muscular arteries, where there's smooth muscle in the wall. And then we get to what are called the arterioles, small arteries, which are nearly microscopic. And then capillaries. Capillaries drain into venules. These are small veins. And venules then drain into the veins, which fuse to become larger and larger until once again we have the vena cava, which empty into the right atrium of the heart. It's amazing how many blood vessels we have. So if, for example, we injected the small arteries and veins with latex, you would see that most of the cells of your body are fairly close to blood vessels. You have an enormous number of blood vessels throughout your body. Actually, if you could take all of your blood vessels, many of them are microscopic, and align them end to end, they would actually wrap around planet Earth about 2.5 times. And so you have an incredible number of blood vessels, although many of them are microscopic or nearly so, as are these small arterioles and venules. The heart typically gets the most attention in the cardiovascular system, but one could argue that in reality, the capillaries are the most important part, because the capillaries are the reason a cardiovascular system exists. The heart simply is the structure which can pump blood to and from the capillaries. But it's here that oxygen leaves the blood and carbon dioxide enters. It's here that the nutrients from your last meal leave the blood and go towards cells, and waste can be eliminated. And so it's these capillaries which form these branching networks these are arguably the most important part of the cardiovascular system. They have little rings of smooth muscle called precapillary sphincters, which can open and close and then determine whether or not or blood services these cells. So there are cells all around these various branches. And so it is at the capillary where blood is getting the cells what they need, and then removing waste from these cells. There are various types of transport here, so materials can diffuse out, pressure can push water and anything that dissolves in it through spaces between where the capillary cells touch. And so once again, capillaries are very important, and this is an important site of transport, and my videos will walk you through some of the basics of that. The blood cells and the blood proteins stay in the blood, but water, gases, sugar, amino acids, etc., they can leave. Now, as blood is coming back through the veins, the problem that veins often have is that their pressure is so low. And to help the blood return from the veins have valves, just as the valves of the heart stop blood from going the wrong way. The valves in veins stop blood from going the wrong way. These valves open when blood is trying to go the right way, and they close when blood is trying to go the wrong way. There is skeletal muscle around these veins, and so that when we stand, for example, every now and then the skeletal muscle around these veins is contracting, and this applies pressure to move the venous blood closer to the heart. So we can regulate whether this smooth muscle in blood vessels is contracting both through neurons. So your brain has centers which control nerves which involuntarily cause vasoconstriction or vasodilation. And then there's also a number of hormones, such as renin and angiotensin, which are hormones which affect blood pressure, or epinephrine, which can cause this muscle to constrict. Obviously, the cardiovascular system, its blood vessels, et cetera, incredibly important. One of the reasons is that cardiovascular disease, which can lead to heart attack and stroke, that is the #1 cause of death in the United States and in the world. And I have videos in other playlists which go through the process by which lipid plaques can start to develop in the walls of blood vessels, which then start to block the blood vessel, making it harder to transport blood through them, and then making it easier that a clot might form and block the lumen here. If this happens in a blood vessel in the heart, that would be a heart attack. If this happens in the brain, this would be a stroke, and it can happen in other organs as well. Just some final comments. While the focus here has been on how the human heart works, obviously we could consider other animals as well. If we start with the most primitive vertebrates, the jawless fish, they have one single atrium and one single ventricle. But then there are two additional chambers. There is a sinus venosus, so there's a chamber prior to the right atrium, which receives blood. And I'm sorry, fish only have one single atrium. And then after the single ventricle, there is another chamber called the Truncus Arteriosus or the Conus Arteriosus. So fish have a 4-chambered heart, but 2 of the chambers are chambers which do not exist in adult humans. So the sinus venosus does not exist in adult humans, nor does this conus arteriosus. And so blood in fish hearts goes from the sinus venosus to the single atrium, to the single ventricle, to the single conus or truncus arteriosus. By the time we get to lungfish and then into sharks, my fault. I apologize, I'm reading up here. By the time we get to lungfish and then amphibians, the atria is then split into left and right atria. And the advantage of this is now that there are lungs, then there are two different kinds of blood. There is oxygenated and deoxygenated blood. So obviously mixing them in one single atrium or ventricle has its disadvantages. And so starting with the lungfish and the amphibians, we start to then see the split of the one single, sorry, this is still the shark, the single atrium into separate left and right atria. And then mammals and birds, this evolved separately in mammal and bird ancestors, then also then split the ventricles so that there is a separation between not only the left and right atrium, but the left and right ventricle as well. In reptiles, there is still there's only a partial septum which divides the two portions of the ventricle, which is still one single chamber. And so hearts evolved over time, and mammalian embryos, like human embryos, still have a sinus venosus, but this will then attach to the wall of the right atrium, which is why the SA node has its name starting in the sinus venosus and ending up in the wall of the right atrium. And then the conus or truncus arteriosus, that will then split to become the aorta and the pulmonary trunk. And so studying, the human heart or human blood vessels, one of the fascinating things that we can then do is consider, the other vertebrates and consider then how it was that the human form evolved over time. And so this was a quick synopsis of blood, hearts, and blood vessels, trying to highlight some of the points made in these videos, but obviously if you would like to pursue any of these points further, each of these videos then can address these topics in greater detail.